Caitlin is our next speaker with ocean warming effects on halibut larval recruitment in the Southern California Bight. Caitlin.
Hi, everybody. My name is Caitlin. I'm really excited to talk to you today about my capstone, which, like Corey said, I investigated the relationship between ocean warming and California larval halibut recruitment. So historically, the waters off California have been supported a really great variety of fisheries for finfish and shellfish, both commercial and recreational. So the cumulative importance of these industries in terms of revenue is like really difficult to quantify. But in 2013, NOAA tried to do that a little bit. They came out with a study where they were trying to estimate the economic impact of commercial fisheries on local economies from 2010 to 2012. So that's 3 years. And they found that the 3-year average, taking into account harvest revenue, output, value added, and total income, generated over $300 million. Not to mention the thousands of jobs that this created scattered across 15 counties. And the study only took into account the California National Marine Sanctuaries, which they stated is really only a third of the total income from commercial fishing in the state. They also made a point to say that it declined every year. In the total catch declined every year. So this study was only looking at those 3 years, obviously, but the trend of declining catch has and continues to be a theme along the entire coast. The South Coast, which is where we are, was previously considered the tuna capital of the world. It's definitely not that anymore. The commercial tuna fishery here is much less active than it has been in the past. And although it still supports some fisheries like squid and sardines and spiny lobster mostly, so many of the previously thriving fisheries have seen collapses or shifts. There's been a lot of shifting. Probably the most well-known example of this is the infamous and elusive white sea bass. So it's been nicknamed this because the gray ghost, because not only how hard they are to catch, but also how hard they are to find and have been for a while. So in the mid to late 1900s, white sea bass populations took a severe decline. So much so that the state legislature passed a law that led to the creation of the Ocean Resources Enhancement and Hatchery Program. So this program is owned and operated by Hub SeaWorld Research Institute, which is the organization that I partnered with for my capstone. And the original program was meant to study both California halibut and white sea bass, but eventually kind of exclusively focused on the white sea bass because limited funding and also at the time just concerns were mounting over how seriously threatened that wild stock was. So they kind of put halibut on hold for a while, but the initial program was meant as an experimental hatchery program to investigate the feasibility of using cultured marine fin fish to successfully enhance wild fish populations that hadn't really been done before. So although measuring the health of adult fish populations is really very, very challenging, by a lot of metrics, the white sea bass seem to have rebounded. And to date, the Institute has 11 grow-out pens all along Southern California Bight, and they've released over 2.5 million fish for replenishment. So the program is the only one of its kind on the West Coast in North America, and it's been established as a model for stocking and aquaculture programs worldwide. So although the halibut got overlooked a little bit in the beginning of the program, the halibut is still a really high value local species that supports a commercial and a recreational fishery here. And because the species has been classified as a good candidate for both food production and stock replenishment, the Institute kept up research on them a little bit, which is very fortunate because as you can see from this graph, halibut populations have been declining since the 80s as well. So the California Department of Fish and Wildlife says that it is one of the most important recreational and commercially fished species in the state and has identified the species as a high priority fishery for scaled management attention. So in 2014, the Institute began culturing halibut to study just the best practices, get a feel for how they can do that, and potentially support the wild stock like they did already with the sea bass. But funding for this is really very limited. So they only did a couple actual releases and really there's no methods of measuring the success of what those programs are. So progress on that has been kind of halted, but this is what one of those looked like. all of that kind of brings me to the motivation for my project and where I picked this up. While culturing halibut, the scientists at the Hubs Research Institute discovered that this species has something called temperature-dependent sex determination. So that means that when larvae are reared in anomalously warm conditions, masculinization of the population occurs. So where a normal sex ratio is about 50-50 for this species, in warmer temperatures, resulting juvenile populations are far more male than female. So based on this information, I was really interested to see if this trait had been exhibited in wild populations and what was going on with wild stock halibut. So my hypothesis was that warmer ocean temperatures would be impacting the local halibut populations, either by forcing migration, so like the halibut would leave, which obviously would affect and shift fishery management, or by staying and actually experiencing the warmer temperatures would increase masculinization and potentially disrupt the sex ratio of these local populations. So I was hoping that I would be able to measure this. So if warmer temperatures are negatively affecting the populations, I used larval recruitment and landings data to see if there'd be changes over time, because there's, like I mentioned, really no way to go out and measure the adult fish. So larval recruitment is data that I had, and landings data is somewhat data that we have. So to look at larval recruitment, I used CalCoffee, which is the California Cooperative Oceanic Fisheries Investigations, which has used cruises like this pattern here to amass a very large suite of physical, chemical, and biological data. I was looking at the biological stuff. It's since the 1950s. So for my purposes, I looked at the larval recruitment of halibut for the entire length of the data set, 51 to 2020. Ultimately, though, I had to, because of the kind of analyses that I was running, and I had to regularize the data, I ended up having to make multiple data frames. So some of these kind of jump back and forth between 1951 and 1984, because in 1984 is when the sampling methods were standardized to these core stations that you see up here. So the gray dots are representing the 66 core stations that have been consistently sampled every single year since 1984. The red dots on the right are scoose stations. So those are 9 additional ones that were added in 2004, which together now make up the current 75 standard station. But prior to that, they were a lot more variable, so it was really hard to standardize. So most of these moving forward are from 1984 to 2020, and I had to take out a lot of the scoose data because it just wasn't long enough. To quantify recruitment, I was taking the annual total sum of the larvae counts per year to keep the resolution of my temperature data. So moving forward, I'm going to show you a couple scatters and a few line plots just to visualize. This is the only time I'll show you the whole data set just because I have it and it's interesting to look at. You can see that oh yes, perfect. Prior to 1984, the data is a lot more variable, which is just reinforcing that I kind of had to use the shorter time series for most of my analyses. So moving forward, you're only going to see 1984 to 2020 instead of this whole thing, unfortunately. But on the right is a heat map just to visually represent the distribution and density of the larvae. So this is at the 66 core stations. This is obviously a sped up video, but I'm scanning through each of the years that I have data for. And you can see that they're really concentrated along the coast, which makes sense, and that tracks because California halibut spend their larval and juvenile stages in shallow water basin estuaries, as Tom mentioned, actually, thank you. And then they migrate into deeper waters as adults. So for my temperature data, Caitlin also did a wonderful job explaining this. I used the same very robust and reliable data set from Scripps Pure because they have taken measurements every single day since the 20s. They actually have taken sea surface temperature measurements at three places, but my specific area of interest happens to be regionally located right near La Jolla. So I trimmed the data to match 1984, which would be right about here where it starts to have that spike. And I'm using annual average sea surface temperatures from La Jolla moving forward as my temperature information. So again, scatters just to see trends. This is showing the 66 core stations from 84 to 2020. And on the left, you can see sea surface temperatures with a moderately positive correlation, 0.43. And as we know, obviously the annual sea surface temperature has been increasing over time because of climate change. So the right, though, is unadjusted annual sum count. So this is just like the data that it spit out when I put in my raw counts. I wanted to draw your attention to these two outliers here. So the top one is 124, which is far and away greater than the next closest point, which is 55. And then that's still much higher than the majority or all of the other data points. The next closest is 37 in 1998. So I had to go back and look at these. What's interesting about these 2 points is that they occur in 2004 and 2005, which, if you remember the scoose lines I mentioned, those were added in 2004. So I kind of wanted to see if there was something going on with that. When I went back to look at the stations that had recorded super anomalously high counts, they were located right next to those scoose stations that were added. So what that means, I don't necessarily know. It's really hard based on the data the data that I have access to know. Pretty impossible to know if it's actually happened. I mean, it was two years back to back that had really high counts, or if it was just an artifact of the data collection and changing of sampling methods. So I ended up just doing everything twice, doing outliers included and then the outliers out. So I'll show you both of those versions in a second. But if I remove those two outliers, then this is going to give me a negative, much more negative correlation at 0.21. So it seems, if this is accurate, that the overall larval counts may be actually decreasing over time. So in addition to the trends with the scatters, I also wanted to look at data as time series. So in the graphs that I'll be showing in the next couple of slides, blue is going to be larval count and temperatures in red. And we wanted to run cross-correlation functions to look for any lags that might be present, right, with temperature increasing larval count at later years. And none of that really returned any biologically significant information, which actually looking at the data kind of makes sense, because if you look over here, you can see that although they're not super strongly correlated in magnitude, obviously, they seem to peak and fluctuate in tandem. So I wanted to overlay them on the same scale to kind of get an idea of what that looked like. And after I adjusted for scale, it seems like that might be indeed the case. This is with the outliers in, right? You can see that really high point right here. If I take them out, then they look to be even more closely correlated. So in summary, contrary to my hypothesis, warmer waters may correlate with increased larval recruitment, at least in the short term. So that's what that previous graph was showing. Like years with high sea surface temperatures also had higher than average larval recruitment. But if I bring it back to the earlier graphs that I looked at, the overall trends show that average sea surface temperatures increase over time, but then larval count is decreasing over time, which is kind of contrary. So, are both of these things possible? What does this mean for wild populations? And then how can I put that in context? So luckily for me, the research institute that I partnered with came out with a study this year, actually while I was working on this project. And they were further diving into how temperature affects halibut larvae and the masculinization. So it kind of helps put some of this into context. They were looking at the thresholds for masculinization, and they found that at 59 degrees Fahrenheit, when the larvae are reared in that temperature, it's about 50-50, so 49.9% are male. When the temperature is 66 degrees Fahrenheit, it is 74.5% male. And then when they were at a high 73 degree Fahrenheit temperature, almost all of them, 98.2% of the population turned male. So what you can see, if I bring that temperature plot back, is that this is very biologically relevant already, because in these years right here, I mean, we've already reached an annual average sea surface temperature that is exceeding 66 degrees. So it could be possible that this is already happening in the stock populations. Additionally, so this is also from that study, but I've also found other research that kind of suggests that the higher temperatures might not actually be dramatically impacting the survivability of the larvae. So they found that in these trials, temperature had a much higher effect, much greater effect on the sex ratio than it did on larval survival. So you can see that the increase in males from 59 to 73 degrees is about 48%, whereas the decrease in survivability, the change in survivability at the same temperature difference is only about 15%. So kind of meaning if it might be possible that in warmer temperatures, larvae are surviving relatively fine, but then they're getting that exposure and the resulting juvenile and adult populations may be seeing the downstream effects of that with a disrupted sex ratio potentially. So lastly, landings data, like I said, is hard to come by. It's also often really difficult to interpret, but it's kind of good to look at for context. Graphs like this one might kind of reflect some of the changes in the population that we're seeing, or just regional shifts in the fisheries, like the migration I mentioned might be happening. We have really no way of knowing what's going on with the adult fish and measuring those populations. But I thought this graph was super interesting because it shows the total number of halibut caught, not just like the total weight or pounds, which it usually does, which brings up a compounding issue of catch size limits and regulations. So female halibut are more often targeted because they grow larger and faster than the males. So you can imagine a world where if there are already fewer males in the population and we're systematically removing the mature, larger adult females, this could bring more disruption in the population dynamics. So in conclusion, to put a lot of this together and give it some real world context, many California fisheries are at risk due to climate-induced influences. We know this, not just the halibut. This is a problem because fisheries represent hundreds of millions of dollars in revenue, thousands of jobs, and really a non-trivial source of protein and food for an ever-growing population in the state of California. And as environmental impacts worsen, increased regulation and really valid conservation concerns and efforts will lead to even further downsizing of the catch and increased pressure on these fisheries. So more research, of course, is needed to look at this species, at halibut, but also other finfish that are promising candidates for replenishment and for aquaculture. But as it stands right now, pretty prohibitive policies exist in the state of California and really affect fin fishing, aquaculture, and especially mariculture practices at the moment. So a reevaluation and kind of updating of some of those laws will probably be necessary in the light of the current climate crisis and the impact that it will inevitably have on our state fisheries. I know it was long. Thank you. I also want to say thank you, Corey, Hannah, and Mark. Thank you for your guidance. And then my chair, Dr. Danielle Halsey at the Institute, was really, really helpful and supportive. Yeah. Thanks.
Two outliers the outliers that you threw out that were very high, did you have any idea what caused those?
I don't have any idea. I might have an idea. I'm not sure. So when I looked, because the data, I can see which specific stations at what time recorded these high counts. I looked at them in ArcGIS, like I visually pulled them up right here. And this might be too far to go back and forth between the slides, but they are also, if you remember the plot of the stations and those red ones along the coast, those were the SCOOS stations that were added starting in 2004. Which is the year that we had the really, really anomalously high counts. And these stations that are recorded the high counts are right next to those SCOOS stations. So even though I threw those stations out of the data analysis, I don't know if they had a different, maybe they changed their sampling techniques, maybe they had different people. I don't know enough about the actual data collection process to know what could have happened in that. But 124 when you're usually getting like 20 is a lot. So
do you think those are unlikely, actually, or recording method or whatever?
I think 124, like, certainly. Yeah. And then this one, I think also because they're back-to-back years, I have a suspicion that when they added new stations and updated the station like pattern, that maybe something. I also know like it's hard to do larvae counts because they have to identify them. They bring them back to the lab and all that. So maybe if they changed, like if they outsourced to Scoos and they started identifying that year when they had other people doing it prior, maybe that had something to do with it. I don't think anybody's looked at that, so maybe you can call attention. Yeah.
Thank you.
Yeah.
You had a line graph
Yeah.
pretty early on showing Northern California from 2015
Ye-
to 16.
yes.
Going way up in the landing.
Yes.
Do you have a theory about comparing that to
Yes. I knew someone was going to ask me about that. So the fishery, so halibut used to be a far more important, like economically. Here in the 70s, 80s, it started doing very, very poorly, but either populations shifted or something else happened because northern populations seemed to be doing a little bit better. So yeah, I was wondering if somebody was going to ask me about that. You're talking about this one. Northern California and Southern California halibut stocks are just completely different and distinct. So they do different things. I mean, obviously right here they look similar, but historically speaking, they operate very differently. I don't know. I mean, that was kind of part of my hypothesis is either they will, like these fish will move if they can't take the temperatures, but it seems like they can. So if they stay, then they're going to be experiencing that masculinization. I didn't specifically look at any of the long-term trends with the Northern California populations. I just know that they are doing far better up there than they have been here. Does that answer your question?